Welcome to the class of comparative genomics. Dear students, now we will study neutral evolution and natural selection. In the last class we have studied comparative genomics approach. So there are two approaches. One is prediction of orthologous genes and another one is prediction of paralogous genes. So there are generally two types of genes. So orthologous genes are present in the different organ, that is the same gene that is present in the different organ. So that are also known as homologous genes, and we use the sequence alignment method. Like using the BLAST we can predict the homologous genes and orthologous genes. Okay, and using the cluster W also you can predict the conserved motif that is shared among them. Okay, so all the orthologous genes contain the conserved motifs. Paralogous genes have been arisen by the gene duplication event that is followed by the sequence divergence. So, and paralogous genes we can predict using FASTA tools. Okay, now today we will talk about the neutral evolution, because so we can also predict the neutral evolution event using multiple sequence alignment method. So first we will understand what is neutral evolution. So neutral evolution or natural selection. So this plays an important role in the natural selection process. So what is natural selection? A process by which biological populations are altered over time as a result of the propagation of heritable traits that affect the capacity of individual organisms to survive. So during the natural process or evolution, biological populations are altered over a time period, and during this process they improve their heritable traits also. So it affects their capacity, because this provides the adaptation also, so that they can survive in the environment. So that the organisms can be adapted for their different environment if there is a changes in the environment. So there was a theory of the natural selection proposed by the Charles Darwin, Alfred Russel Wallace in 1858. Okay, and more obscure formulation had been arrived at by earlier researchers. So what Darwin said? So Darwin suggested that there will be the within the same population there will be the two types of the two types of category. There will be two types, so we can divide that population: those who can survive, those who will be adapted to that condition, and there will be some organisms that will not be adapted to that condition, so they will not survive. So there is the rule of the survival of the fittest. Those who are fit, they can survive on that condition, during that condition. So that was the theory that was suggested by the Charles Darwin. So in contrary to the Lamarckism, so Lamarck suggested that organisms try to adapt that conditions, and there is the evolution in that process. They change their gene and they adapt it to the condition. So it happens in the next generation, doesn't happen in the same. Okay, so gradually there is a change, because within the same generation there cannot be any change in the gene frequency. So in the next generation the change will be appeared, so next generation will be improved. So by using our organ more frequently in that condition, that will improve our gene condition also, gene content also. Okay, so that was the theory of the natural selection that is based on the evolution. Now there is the neutral theory of the evolution. What is this? Neutral means it doesn't affect the protein or organism. Vast majority of molecular differences are selectively neutral, because there is a change in the gene frequency, there is a change, but it doesn't appear in the protein structure, doesn't appear in the organism. Okay, so these genome features are neither subject to or nor explicable by natural selection. So although these changes are hereditary in the next generation also, but it doesn't affect the organism functioning or structure. So most evolutionary change is the result of genetic drift acting on neutral alleles through drift. So these new alleles may become more common within the population. So the allele will become more common within the population, but it will not affect the structural function of the organism. So they may subsequently decline and disappear, or in the rare cases they may become fixed, meaning that the substitution they carry become universal feature of population. So it will be there in the gene, okay, but it doesn't affect the structure function of the species. So you can relate it to the pseudo genes also. Sometimes genes do not function, but it reside. But whenever there is this condition, the pseudo genes can also become active. So in that way they can play an important role. The pseudo generally it is related to the pseudogenes, but pseudogenes are I think pseudogenes become active when we get the disease or any unusual circumstances. So gene can play an important role during that condition. So these are the significance during the unusual circumstances whenever we get the infection also, but it is not given here. Okay, so this is our way of thinking. Some of there are some scientists those who suggested the pseudogenes played an important role in the during that disease process, or sometimes they become active during the evolution process. Neutralist selection is debate, so which is prevalent evolutionary form. So this is hereditary. The changes transfer from one generation to another, but there is no evolution. Okay, although the genes pseudogenes are also transferred from one generation to another. So using the comparative genomic tool we can identify those mutations, like assume two closely related organisms Ka and Ks. So purpose of that probability of a back substitution means X can be changed into A, or A can be X can change into A are unlikely. Example, mouse and rat, human chimpanzee. So K of coding based substitution result in amino acid change, and Ks of coding based substitution that do not result in the amino acid change. Okay, so it might happen. K divided by Ks, measure of evolutionary constant. K divided by Ks, if it is less than 1, the strong purifying selection. If it is greater than 1, the possible adaptive or positive selection. Means if there is a coding sequence substitution that result in the amino acid change, so that can be divided by the Ks. Okay, so if it is more in number, then the ratio will be greater than 1. If the substituted base sequence that result in the amino acid change, means there is a change in the protein also, so there the ratio will be greater than 1, means the number of the base substitution that do not result in an amino acid change, that is less. Okay, and if we calculate the ratio, so that will be greater than 1. So this is the possible adaptive or positive selection, in which we have the change in the protein, and sometimes there is no change in the protein also. If the Ka divided by Ks, the ratio is less than 1. Okay, this is a strong purifying selection. Comparison of mouse, rats, chimpanzee, human chimpanzee, that is 0.20 Ka divided by Ks, Ka divided by Ks, mouse and rat. So the ratio is 0.13. The difference attributed to relaxed evolutionary constants. 4.4% human chimpanzee orthologs have Ka divided by Ks greater than 1. So this is the ratio for human and chimpanzee. So this can be hypothesized that to be the under positive selection. genes that are involving reproduction. So we can do the same species comparison also. HapMap project. So multi-country effort to identify the catalog genetic similarities and differences in human beings. So we can compare the human of the different countries. So the genetic data are being gathered from 4 populations like African, Asian and European ancestry. Okay. So and that have been analyzed. So SNP we can also analyze. We can do the comparison. SNP, single nucleotide polymorphism. So there the second generation human haplotype map has over 3.1 million SNPs. So there the change like single nucleotide polymorphism. Okay, so by doing the SNP analysis we can identify this will be changed. Infinite site mutation model. Under infinite site mutation model, it is given by Kaimura in 1969. Mutation never occur twice at the same position. Okay, but it may happen. This the condition is equivalent to the perfect phylogeny. So sometimes it doesn't happen, sometimes it happens. So this is a particular case. Recombination play an important role in the evolution also, because the chromosome is divided into parts and this part can be exchanged, and this is responsible for the variation in the different organism. Discovering recombination events. So not infinite mutation site junction is equivalent to perfect phylogeny. So there is the gamete test. Under infinite site mutation model, there must be recombination event between these SNPs. Okay. Haplotype, genotype and phasing problems. So haplotype, description of SNP allele on a chromosome. A genotype, description of allele on both chromosomes. So these are the different types of, these are the definitions that has been given here. Phasing the process of assigning haplotypes given genotypes. So here you can see 2 haplotype per individual and this is a genotype. So there can be exchange of the part between these. Linkage disequilibrium, the non-random association of allele at 2 more loci. So two alleles can be associated with each other that are on the different locus, but they can be associated with each other. Haplotype frequencies deviate from the values they would have if the genes at each locus were combined at random. So there will be strong disequilibrium, low recombination level. So in this figure you can see, in this table you can see, so these are the different recombinants. Haplotype frequencies are given here and allele frequencies are also given here. Capital A, capital B, small a, small b, and the recombinants are also given here. You can see the difference. Expected and observed frequencies are given here. So this is the example of linkage disequilibrium. So haplotype frequency deviates from the value they would have if the genes at the locus were combined at random. Okay, now this lecture is over.